welcome to every one of you. We're very happy that you appeared in this huge number. So my name is Julia, and I'm the ESID country representative of Germany, and I'm very happy to moderate this journal club, ESID Junior Journal Club session today. And before I introduce you to our today's session speaker, I want to start with some organizational details. First thing is, if you have any scientific questions during the talk, please write them to the Q&A panel on the lower part of your screen, and we will answer them in a discussion after the talk. If we are running out of time, no problem. If we're running out of time in a discussion, no problem. We will copy the questions and get back to you afterwards. And also at the end of the talk, you will have the possibility to speak up freely if you want to do that. So in that case, please just raise your hands in the Zoom call, and then I will ask you to speak up and Eleanor will unmute you. Also during the talk, you can exchange your own experiences with others or comment on cases in the chat panel. There you can also have one-on-one chats. And also Laura will have 3 poll questions for you that you can participate in during the talk. You will see that, when it comes to the poll questions. But if you have no further organizational questions for now, then I'm very happy to introduce you to Laura Batlle Masó. She is a postdoctoral fellow at the Hospital Vall Hebron in Barcelona, and she's working with Dr. Pere Soler and Dr. Roger Colobran. Actually, she has a biology degree, and she specialized in genetics and bioinformatics during her PhD. And the main line of her research now is to expand the knowledge on genetics underlying inborn errors of immunity. So for that, she has been working quite a lot with somatic variation. And she has a huge interest in the use and development of bioinformatic tools to provide a molecular diagnosis to patients with inborn errors of immunity. And she says about herself that she's a bit new in immunology, but very eager to learn. And yeah, all right, so I'm very happy to hand over now to Laura, and I'm very excited for an interesting talk about the detection of somatic variants in autoimmune lymphoproliferative syndrome. Please enjoy.
Thanks a lot, Julia, for this nice introduction. I will share my screen. Okay, so hello everyone. Thank you so much for joining us today. And thank you to the ESID Juniors for inviting me to participate in this journal club sessions. So today I'm going to talk about this recent paper that we published, which is entitled Detection and Evolutionary Dynamics of Somatic FAS Variants in Autoimmune Lymphoproliferative Syndrome, Diagnostic Implications. So first of all, I wanted to start doing a bit introduction to ALPS. ALPS stands for Autoimmune Lymphoproliferative Syndrome, and it's mainly characterized by chronic inflammation, chronic lymphoproliferation, lymphadenopathy, autoimmune cytopenia, and increased risk of malignancy. There are 2 main required diagnostic criteria for ALPS. And these are chronic, more than 6-month, non-malignant, non-infectious lymphadenopathy or splenomegaly or both, and elevated double negative T cells, which we'll talk about in a bit. There are also accessory criteria such as defective lymphocyte apoptosis, somatic or germline mutations in FAS, FAS ligands, or caspase-10, and also other markers such as elevated plasma levels or EL10, EL18, vitamin B12, immunohistological findings, autoimmune cytopenias, elevated IgG levels, and of course, positive family history. As Julia said, I'm not a clinician myself, but just a few highlights for therapeutic approaches. ALPS patients are usually treated with corticoids initially, and then depending on the levels of the disease, the severity of the disease, they move to immunosuppressants such as sirolimus. And finally, in the most severe cases, they go to hematopoietic stem cell transplant, which is the curative strategy. I wanted to just give an overview of the molecular basis. So ALPS is a defect in apoptosis, and apoptosis is crucial to maintaining homeostasis and controlling self-tolerance. There are two apoptotic pathways, the intrinsic pathway and the extrinsic, and we're going to focus on the extrinsic one today. This pathway is mediated by FAS and FAS ligand, and as we can see here a bit more on data, both FAS and FAS ligand are homotrimers that initiate the apoptotic signal with the help of other molecules such as Casp8 and Casp10. And here we can see this very important intracellular domain, which forms this death-inducing signaling complex, also named DISC. Let's enter now to the genetics part. Genetic defects in the genes encoding the proteins involved in the extrinsic apoptotic pathway are responsible for ALPS disease. And in this table, we can see the most common germs. So germline homozygous mutations in FAS, germline heterozygous mutations in FAS, somatic mutations in FAS, which are the main start of this journal club. But also we have mutations in other genes such as FAS ligand and Cas10. Unfortunately, there is still also this last group, which is the patients that after a first genetic approach remain without diagnosis. Let's focus now on the somatic ones, and here we can see this figure, which is basically a representation of the FAS protein, where we can see all the somatic variants that have been described. There are different types of variant, frame shift, missense, nonsense, splicing, and also this in-frame deletion. And we can see that they are all grouped at the intracellular region of the gene, especially at the dead domain. Here, I just want to stop for a sec and briefly introduce somatic variants. Although often used as synonyms, mosaicism and somatic variants are not the same thing. Mosaicism can be inherited from our progenitors, and if it's located at the gonads, so here we have gonosomal and gonadal mosaicism, and on the other hand, somatic variants are the ones that appear during development, and they can be restricted to a group of cells if they appear later in development, or affect the whole individual if they appear in very early embryonic stages. So in ALPS, these somatic variants that we're looking for are located at DNT cells, which are cells CD3 positive, but CD4 and CD8 negative. And these cells in healthy individuals, here we have a control and a patient, should be less than 2.5%, but we can see that in the patient here in this control, in this quadrant, we see an elevated number of these cells. I don't want to enter in the dynamics of the formation of these cells, but here just this figure is really nice, and I really recommend you this paper, which is the first one who describes somatic variants in ALPS. So yeah, basically we have seen that these double negative T cells are just a very small fraction of T cells. So if we have a somatic variant in these double negative alpha beta T cells, in all of them, it will only be seen in a small proportion of T cells, and even a smaller proportion of whole blood, because the frequency keeps getting diluted. So one of our objectives here for genetic studies is to try to enrich this sample that we use for our studies. And we can do that with different strategies. We can separate T cells, and we can do that with magnetic beads of these centrifugation methods. And we can also separate specifically these DNT cells by sorting, which is a bit more expensive, but will be very robust. So here's the first concept I want to really stress, and is that having our starting sample, it will depend really if we are talking about both DNTs or T cells, because this will really affect the result in terms of variant allele frequency. Also, another important concept, I've talked briefly about treatment. When we start giving our patients immunosuppressants, this number of DNT cells decrease dramatically. So if we study the DNA in a post-treatment sample, we will have only few DNT cells, and the variant will be quite difficult to detect. So in genetic studies, usually it doesn't really matter if we have a sample when the patient is under treatment, but in this case, it really matters. So that's an important concept. Let's move now to the detection of these somatic variants, and we'll start from the beginning. Sanger sequencing, one of the most common ways of sequencing for small regions. And here in this paper that we published recently, we show this figure. This is not ALPS, this is chronic granulomatous disease. And we can see 3 generations. In the first generation, the grandmother, we can spot this somatic variant that is seen as this small peak. And here, through NGS sequencing, we can calculate variant allele frequency. But of course, in this Sanger sequencing chromatogram, we are not able to assess the frequency of the variant. In the next generation, the variant was inherited as heterozygous, so we see both peaks are more or less the same height. And in the third generation, we see that the variant is homozygous. So here we have a nice example of how to visualize this somatic variant. And here this image is quite nice, but this can be actually quite of a challenge, because if we have a noisy signal, these small peaks can be easily confounded with background noise. So Sanger sequencing can be useful to detect this type of variation, especially if we're studying a very specific region, but also come with some disadvantages that we should take in account. So moving to next generation sequencing, I'm going to give you here an overview of the whole process that we use for detecting somatic variants. And yeah, I'll try not to be very bioinformatician here, but yeah, basically, when we perform sequencing, even target sequencing, exome sequencing, whole exome sequencing, we obtain the data from our sequencer, and then what we have first is a FASTQ file. This file contains all the reads that have been seen in our sample, and we can align that to the reference genome. Once we do that, we obtain a BAM file. And BAM files are very, very useful because they can be visualized at software such as IGB. I don't know if you have ever used that, but it's very useful because you can see all the reads and the reads that contain your variant. So that's quite an important thing when we are looking for somatic variants, especially if we don't have specialized colors, because we can go back to the BAM file and visualize there our variant. BAM files are also the starting point for callings. And usually what we do is germline variant calling, which will give us a BCF, which is a variant calling file. So basically a file which contains a list of all the variants that are different from our sample regarding the genome of reference that we are using. This is like the standard protocol, but it's important to know that added to this germline variant calling, we can perform other types of calling, such as structural variant calling or somatic variant calling, which we'll see in a bit. And basically, all types of callings will give us these variant calling files with all the variants. And from there, we can proceed to annotation. Annotation will give us all the information that we need from our variants, so in which gen they are located, population frequency, conservation scores, and so far, so on. So these annotated files are usually converted to a user-friendly format, such as an Excel file. And here we can start the filtering process. So basically, we filter out the variants that we're not interested in, and we try to find the variant that is causing our disease. I'm pretty sure that you are familiar with parts of this pipeline or even the whole thing, but I think that now we'll move to the specific somatic variant calling, and I thought it was nice to give an introduction of that. So somatic variant callers, what we do is basically to spot these variants that are not picked up by germline variant callers. And they do that by evaluating allele frequencies. So variant allele frequency is the percentage of reads that we have with our variant versus the total amount of reads that we have in a specific position. And in this paper in 2019, what our colleagues saw is that we have, of course, germline variation that can be homozygous, so variant allele frequency will be 100%, and heterozygous, here we have these box plots at 50% more or less. And for somatic variants in inborn errors of immunity or primary immunodeficiency, they show that variance can be around in between 2% and 40%. So we have a wide range of different frequencies. We can evaluate different colors because, of course, many bioinformatic tools have been developed during the years to pick these variants. But here we have to be a bit careful because somatic variant colors are usually developed for cancer. So they work in a pair mode when you have a healthy sample and then a tumor tissue. So it's important to take in account that these colors may not be super specific for our cases. But here what we did is to evaluate a set of different samples with different variant allele frequencies using a bunch of different colors. And here we can see that in this specific moment with these specific samples, VARScan2 was a color that was really working for us and is the color that we're actually using. But the point that I want to make here is that there are many different colors and many different parameters that you can adjust to call these somatic variants. And you have to just play a bit with the settings to find the variant that we're looking for. So using NGS, apart from these somatic colors from data that we already have, such as target sequencing data or exome data, we can also perform more sequencing. And in this case here, I want to highlight deep amplicon sequencing. In this case, when we have a specific target region, it can be a whole gene or even a small amplicon, a small region, what we can do is to design an amplicon. So basically, we pick our region of interest, we design the primers, and then we sequence a lot that region. This will be very helpful because the more time we sequence the region, the more are the chances that we'll see reads supporting our somatic variant. This can also be done in a more genome-wise gene using, for instance, single molecular inversion proofs, which are a very nice approach that we're exploring now with our colleagues at Vall University. Okay, so this led that to the end of the introduction. And basically, I want to highlight these two concepts. So how to increase the likelihood of somatic variant detection. We can do that by improving our sample using these enrichment protocols to isolate CD3 or even DNT cells, or improving our technology, such as specific somatic colors or higher depth sequencing. Let's move now to our case of study in the paper. So our patient was a 1-year-old boy who presented with splenomegaly, elevated DNT cells, as we can see here, these images that you have already seen, and also elevated vitamin B12, EL10, erythema, persistent cough, and specific fever, and for all of that, a clinical ALPS suspect. So this brings me to the first question, which is, which could be the first genetic study to be performed in this case? A, Sanger sequencing of the candidate genes, B, whole exome sequencing, or C, deep amplicon sequencing of candidate genes. And please bear in mind that this was 2017. Eleanor, if you can, please open the poll now. Okay, so many of you have both Sanger sequencing, and that was indeed what we did, because first, at that time, Sanger sequencing was the gold standard using in our lab, and second, we had very specific candidate genes, so it was not a huge effort to really sequence these candidate genes. For sure, it was more cost-effective at that moment than doing a whole exome. So here we have highlighted the correct option at that time. So we went ahead and performed Sanger sequencing from whole blood with a sample that contained these 7.5 double negative T cells, but unfortunately we were not able to see any mutation in any of the candidate genes. Here you have an example of FAS, a region of the FAS gene. However, given the tight relationship of ALPS and double negative T cells, we repeated the Sanger sequencing with a sample enriched in CD3, in this case using magnetic beads. And in this case, we detected this 4 base pair insertion in the exon 8 of the FAS gene. And as you can see, the insertion had a frameshift effect that causes this premature stop coding of the protein. So we consider this as a likely pathogenic, as a good candidate, because we have this frame shift effect. But before moving on, I wanted to ask you another question. So in this case, thanks to Sanger sequencing, we were able to identify this insertion, which we consider causative. But my question here is, do you think this approach could be used to routinely detect somatic FAS variants? A, yes, it seems reasonable. B, no, you were lucky. Or C, maybe, but it's not the best option. So we can open the poll now. Okay, yeah, so many of you say that maybe, but it's not the best option. And I fully agree with you. In fact, this question, all the three answers could be true in some points. So, yeah, it seems reasonable because as we had the opportunity to enrich our sample for CV3, we can again perform Sanger sequencing and see if we are lucky. And yeah, we were indeed very lucky because in this case, we had a frame shift variant, so we can see all these little peaks. But imagine that we had only a missense variant. So just one of these peaks could be very easily mistaken by background noise. So I will say we were a bit lucky. And yeah, for the last option, yeah, I think it's a good option, but in this case, it can be a bit challenging. We were in the ALPS, we have the FAS gene, and somatic variants had already been described there, so it was kind of a good strategy to go for sequencing in this specific gene, but if you have to sequence a lot of genes, this will actually not make a lot of sense. So yeah, we have here numbers saying you were good. Okay, so next, we further studied the variant itself, and we look at it and corroborated that it was not found in healthy population, and it was located at the gene already associated with the phenotype of our patient. Therefore, it is considered likely pathogenic by the American Council of Medical Genetics. And with that, the patient initiated immunosuppressive treatment and has a clinical diagnosis and has been actually quite good since that. However, at the technical level, we wanted to go a step farther into studying this variant. And for that, I wanted to ask my third and last question, which is, what would you do next? You would consider the variant as pathogenic and close the case. You would validate the variant using whole exome sequencing, or you would validate the variant using deep amplicon sequencing. We can open the poll. Okay, so I agree with you. Many of you pick option C, and I think that's actually the correct one, because in this case, of course, we're not going to close the case because we work in research and we really want to know more about the patients that we diagnose. Using whole exome sequencing would be not very useful in this case because, of course, we will have NGS data, but maybe the coverage will not be as deep. And we don't need to analyze the whole exome if we clearly have a candidate. So using deep amplicon sequencing is, I will say, the more efficient option in this case. So we obtained a new sample. This was 2021. And due to the immunosuppressive treatment, the patient at that moment had normal values of DNT cells, at this case 0.89%. And we performed deep amplicon sequencing, or DAS, at very high coverage. And here you can see highlighted the total number of reads, which was over 20,000, very good coverage. And here, the number of reads that were supporting our somatic variants. And of course, this is very small proportion of reads, is just 0.68%. But if we see the other alleles, which will be considered background noise, we can see that there's a significant difference. So we concluded that, yes, we can see our variant here, even if the patient is under treatment. Again, what we did here is to isolate these CD3 cells, and we enrich our sample using a centrifugation method, in this case, the Rosette set, and perform again deep amplicon sequencing. And as expected, at more or less the same coverage, we can see now that the reads supporting this insertion are much higher, and the variant allele frequency has almost doubled. Therefore, we corroborate that we could detect somatic variants in patients under treatment with low DNT counts when performing this targeted search. So if you remember the slide of increasing success, here we prove that enriching our sample really works. In order to explore a bit more this option, what we do is to perform somatic calling in this deep amplicon sequencing data. And here, for instance, we use BarScan, and BarScan gives you the output of course, a lot of calls, and many of them are just background noise. So what we did here is to plot the variant allele frequency of each of these variants that was called versus the genomic position. In this case, the exon that we already knew that was the one containing the pathogenic variant. And we did that for whole blood, you can see here in red, and also for CD3 cells in blue. And you can see that our pathogenic variant is quite easy to detect here because variant allele frequency is much higher than the background noise. So we hypothesized that this could be a nice strategy to apply to another patient, and we're actually working on gathering cohort of previously undiagnosed ALPS patients to try to perform this strategy and see if we have new candidates using this pretty straightforward a strategy. Of course, there are also other methods, as I mentioned, like MIPS or digital droplet PCR, but we believe that this deep amplicon sequencing and then somatic variant calling is also a nice and fast strategy. So to wrap up the description of the case, we took advantage that we have been following these patients for over 5 years to describe the evolution of these DNT counts and the somatic variant allele frequency across time. And as expected, they are closely correlated because both start elevated and decrease dramatically due to the immunosuppressive treatment, here indicating with this yellow box. So this highlights again the importance of recovering pretreatment samples with high DNTs to perform genetic analysis in order to increase the chances of detecting the somatic variants. And here we have again all the counts. If what was important to us here is to perform this in a very standardized manner, so trying to have the same depth of coverage for all the samples. Okay, so this leads me to my last slide. So to recap, I want to stress 3 important points. First of all, that the use of pretreatment samples and CD3's enrichment increase the likelihood of identifying a pathogenic somatic FAS variant. That optimized sequencing and analysis pipelines are needed to find somatic variants with low variant allele frequencies, and that the study of somatic FAS variants can lead to a molecular genetic diagnosis in previously unsolved ALPS cases. And finally, of course, I want to say thank you to all the team who participated in that study, and also all the persons working in our group, and again to ESID for giving me the opportunity to present our work. Thanks.
Thank you so much, Laura, for this excellent presentation. It was very interesting to dig a bit deeper into this, genetic diagnosis making. If you have more questions to Laura, you can certainly connect with her and ask her about her presentation and genetic questions. And I want to use this journal club to remind you of our next ESID Junior clinical case session, which will take place on December 21st this year, so the last one for this year. And I want to invite you to take part in this one too.
Yeah, I just want to say thanks a lot again to ESID and Julia for moderating beautifully this session, and also to all the attendees that if you have further questions, do not hesitate to get in touch through the WhatsApp group, my email, or any way of contact that you may think of. Also, we are very interested in this, so if you have unsolved cases, if you want to implement these pipelines in your center but you don't have a bioinformatician, I can totally help out with that. So I'm very open for collaboration in this regard. Do not hesitate to contact us.
Thank you so much, Laura, for this great presentation and for this great offer and opportunity.
Thank you.